Hello children, today I will be taking the topic DNA fingerprinting in video number 10, and with this topic I will be introducing you to yet another interesting branch of biology called forensic science. What is forensic science? Forensic science is also known as criminalistics. It is a branch of biology which deals with the application of science and scientific method in the judicial system, both civic law and criminal law, mainly on the criminal side during criminal investigation. Samples at the crime site, whether it's murder or rape, some of the samples left behind may be the blood, saliva, semen, hair follicle, skin, nail or bone of the victim as well as the convict. From this sample, the DNA is isolated, and after a sequence of steps which we will be dealing in the last slide, the final product is a DNA profile. And this DNA profile obtained from the sample obtained at the crime site is compared with the DNA sample of the suspect. And if it is found to be matching, the criminal is convicted. So this particular branch is called DNA profiling or DNA typing, but it is popularly known as DNA fingerprinting. Why? It is called DNA fingerprinting because it replaces the conventional investigatory tool of forensic science called fingerprinting. Our fingerprints, we have on our fingers some ridges which forms our fingerprints. It is very unique to an individual and no two individuals have the same fingerprints. And it was used till the 19th century, it was used as an investigatory tool for investigations of crimes like murder, rape, theft, etc. But what happened is that the criminals started becoming wise. They became very prudent and they left no imprints behind in the crime site. They either wore gloves or burned their fingers by dipping in acid. So they were not easily caught. Not only that, the crime rates increased because many people were victims to kidnap, murder, etc. just for their fingers. Their fingers were chopped and the fingers were used for their fingerprints to open their lockers, houses, cars, etc. So forensic scientists were thinking of better options, better investigatory tools. It was around this time, a professor in England, Sir Alex Jeffreys, was doing studies in his lab. He was investigating the inheritance of hereditary diseases in families, and he noticed a certain segment of the DNA which was very unique called repetitive DNA. In that sequence of DNA, a small stretch of DNA was repeated many times. And he found that this particular repetitive DNA, this unique segment, showed high degree of polymorphism. You know what is polymorphism, so high degree of differences between individuals to individuals. But he found that it was inherited from parents. He found that the siblings and the parents had similarities with respect to this unique segment. And this repetitive DNA segment is known as satellite DNA. We will come to that in the next slide. So it was Sir Alex Jeffreys who discovered DNA fingerprinting, and he made a DNA profile in his lab, and this was the world's first DNA profile, and it was used for an investigation. It investigated and the culprit was found guilty. It was a case of the criminal Colin Pitchfork. He is a criminal. He committed 2 murders, but before murders he raped and then murdered 2 15-year-old kids in England, and he denied the charges. But finally he had to give up and was sentenced in 1988 when his DNA sample from his blood was matching with the sample obtained at the crime site. Henceforth, DNA profiling or DNA fingerprinting was used as an evidence in the court of law. Thus, in the field, this was a revolution in the field of forensic science, and the discovery of DNA fingerprinting by Sir Alex Jeffreys was a breakthrough. It trapped many hundreds of killers, murderers, robbers and rapists, freed many innocent people and also revolutionized science and forensic science. So once again, reiterating the principle or the basis of fingerprinting is that unique segment found in the human beings which was different from one another, which showed high degree of polymorphism. That unique segment was called repetitive DNA. In that repetitive DNA, there was a small stretch of DNA repeated several times. And when this was centrifuged in a centrifugation called density gradient centrifugation, this particular repetitive DNA, that small stretch of repetitive DNA was separated from the bulk DNA as small peaks. So you can see with another color I'll highlight small peaks, very small peaks. Okay, these were the repetitive DNA, and the genome, the entire genome took a bigger peak, was a major peak. So it got separated. So in the density gradient centrifugation, you can separate these repetitive DNA from the bulk genome. The bulk genome appeared as a large peak, major peak, whereas the repetitive DNA appeared as small peaks. This is the repetitive DNA. And this repetitive DNA, it looked like satellites. So these were called satellites. And the satellites were divided or classified into mini satellites, micro satellites, macro satellites, etc., based on the composition of the bases, the number of repeats, as well as the length of the satellite. So once again, these satellites were classified into mini satellite, micro satellite, etc., based on the length of the segment, base composition and the number of repeats. And these satellites, the properties of these satellites, if I highlight it in points, the properties is that they do not code for proteins. First property, they do not code for proteins. Then they form a major chunk of the DNA profile. A major portion of our human DNA is made up of these satellites. That's the second point. Third point is, as Sir Alex Jeffreys found, it showed high degree of polymorphism. And he also noticed that we inherit this polymorphism from the parents. It was passed from the parents to the offsprings. So once again, about the satellite DNA, you have to remember these 4 points. First point, they do not code for protein. Second point, they form a large portion of the human genome. Third point, they show high degree of polymorphism, differences. Your satellite DNA will be different from my satellite DNA. So it shows so much difference because we are unrelated. But the satellite DNA of mine and my daughters will be same. Okay. So the fourth point states that we inherit this polymorphism from our parents or from one generation to the other generation. And that is regarding the features of the satellite DNA. And in DNA fingerprinting, which satellite was used as a probe? It was the VNTR. VNTR was a mini satellite. So in DNA fingerprinting, there are different satellites. I told you it's classified into mini satellite, micro satellite, macro satellite. The satellite that was used for DNA fingerprinting was a satellite called VNTR. V stands for variable, N stands for number, T stands for tandem, and R stands for repeat. And this particular satellite belonged to the mini satellite. So once again, the principle or the basis of DNA fingerprinting. The first and foremost point is that there are segments. DNA fingerprinting involves finding the differences in certain specific regions of the DNA called repetitive DNA. In these repetitive DNA segments, there are small stretches of DNA that are repeated many times. And these repetitive DNA can be separated from the bulk DNA in a density gradient centrifugation. How it is identified? The bulk genome appears like a major peak, and the repetitive DNA appears as small peaks. And hence the repetitive DNAs are not called, in forensic science, they are not called repetitive DNA, they are called satellites. That's all about introducing you to satellites. Now the properties of satellites: they do not code for proteins, they form a major chunk of the human genome, then they show high degree of polymorphism, and it is inheritable. And because it is inherited, they form as, they are very good tools as genetic markers. And in DNA fingerprinting, which satellite was used? It was a mini satellite called VNTR, variable number tandem repeats. Now, if they ask for the board exam, what is VNTR? One point you will get is, VNTR is a mini satellite. Second point you can give based on the marks, you can increase. You are going to elaborate what is VNTR. So, you start from the reverse. VNTRs are DNA repeats. So, the word R is there. Repeats that are arranged tandemly, and the number of these repeats, so number varies from chromosome to chromosome. So, once again, if VNTR is R, first you must introduce VNTR is a DNA probe which is used in fingerprinting. So, and it belongs, second point, it belongs to which satellite? Belongs to the class of satellite called mini satellite. And third, you are elaborating. The abbreviation you can elaborate: variable number tandem repeat. You just elaborate that. And fourth, you are explaining what is it. When you are explaining, remember, explain from reverse. So, you will get every keyword. They are repeats of DNA. Then tandem stands that are arranged tandemly, and the number, N stands for number, not a verb there. And the number varies, how? From chromosome to chromosome. So, once again, I hope this slide is clear. It is very important because many questions can be asked. Questions that can come for the boards are, one is a direct question: what are satellites? And looking at the marks, you have to add on points. What is a satellite? What are repetitive DNA? What is VNTR? And one as a whole, it can be asked, the principle or the basis of DNA fingerprinting. So, with three different colors, I have reiterated three aspects here. So, once again, repeat in a nutshell, the principle or basis of DNA fingerprinting is, it involves identifying differences in some specific regions of the DNA called repetitive DNA. And in this repetitive DNA, there is a small stretch of DNA that are repeated many times. This repetitive DNA, talk about the centrifugation. In density gradient centrifugation, the repetitive DNA is separated as small peaks from the bulk genome, and the bulk genome appears as a major peak. So, your first point is how you are going to define repetitive DNA as satellites, and these small peaks were referred to as satellites that I first indicated here. I will give a red color to it. So, first I told about the centrifugation. Then the second is, these satellites can be classified. So, you are talking about the classification. It is classified into mini, micro, macro satellite based on what and all criteria: length of the segment, base composition, and number of repeats. So, your second point was the classification of the satellites. Then third, you are giving the features of the satellite. There are 4 features of a satellite. So, that stands as a fourth point. So, you will remember 4 features. They do not code for proteins, they form a major bulk of the genome, they show high degree of polymorphism, and they are inheritable. And because they are inheritable, they serve as genetic markers. And fifth point you can say is, the type of satellite used in our DNA fingerprinting is which satellite? It is a mini satellite called VNTR. Now, we can elaborate VNTR. What is VNTR? Elaborated variable number tandem repeats. And to explain VNTR, start from the reverse. They are repeated DNAs, tandemly arranged, and their number varies where from chromosome to chromosome. So once again, don't make it too much in your own words, go by the textual words and you can reach the examiner. Very, very important slide. Now moving on to the steps of DNA fingerprinting. The steps are given in the form of these diagrammatic representations so that it will have that visual effect in your mind. Okay. The first step is isolation of DNA. So the first step in DNA fingerprinting is isolation of DNA, and that we will be doing it in the lab. We will be isolating the DNA of any vegetable. Okay, so that step, when it is asked in the exam, just write it only the heading isolation of DNA. The procedure will be doing it in the lab. And this also you have seen, which coincides with Human Genome Project. The first step is the same, isolation of DNA. Second step is also same as in the Human Genome Project, fragmentation. So in Human Genome Project also, the second step was fragmentation. As the name suggests, the DNA, see this entire DNA, human DNA, how long? 2.2 meters long. Okay, that is cut into fragments and it is cut with the help of this enzyme. What's the name of the enzyme? I told you, restriction enzyme. And in biotechnology, we are going to learn there are two types of restriction enzymes, restriction exonuclease and restriction endonuclease. Exonuclease and restriction endonuclease. The type of enzyme, both are scissors, but the type we are using here is restriction endonuclease. So restriction endonuclease is this, these scissors. So the purpose is to cut the DNA into fragments. So we get this. So you can see here fragments are obtained. The next step, if I continue with red, the next step is that these fragments are put in an agarose gel. So this is a gel made up of agarose. Agarose gel is made from agar. It is an agar medium made from algae. Okay. So an agarose gel slab. So you can understand the consistency of gel. It is jelly. Thick gel. Okay, and this is a well. This is a well, a small incision in the agarose gel. Just imagine a slab which is jelly, little more thicker than the consistency of the jelly you make at home, like candle wax. But wax gets solidified, but little jelly like. And there's a pocket or an incision. There these fragments are placed. Okay, once the fragments are fed into the, and this is called, this incision is called, in terms of biotechnology, it's called well. And here there's only one well shown. So in this well, the fragments are fed. And what happens is electricity is made to pass. So this is connected to a wire. Electricity is made to pass. And when the electricity is made to pass, we have the anode and the cathode region. And the DNA we know is negatively charged and has a tendency to move towards the anode. Okay. Now the purpose of gel electrophoresis is to segregate it based on their size. Okay. Now we have fragments of various sizes. So we are going to segregate it based on their size. So I am just showing on this, it's segregated. I am taking yellow to show the DNA fragments. So you can see the big ones are here, but as you see, small ones. So remember, the smallest fragment move further away. The farthest away is the smallest fragment, and the bigger one is towards the anode. Okay, and we know that because DNA is negatively charged, it moves towards the anode. So the bigger one is to move towards the anode. The smallest one, it moves further away. Okay, so the purpose of gel electrophoresis is to segregate the fragments based on sizes. So I'll continue with red color. So the third step is electrophoresis, and I'm specifying gel electrophoresis. So once again, what is gel electrophoresis? We'll be learning in biotechnology once again, but I'm just giving you a superficial detail about it, that it is an agarose gel slab through which electricity is made to pass, and there is a well into which the fragments are fed, and once electricity is made to pass, the DNA gets segregated based on size, and DNA, remember, DNA being negative moves towards the anode, and here you can see the smaller segment moves further away, farthest moves further away. So we got it segregated. Now the next step is simply, if I show you this as a slab, my book as a slab, the gel slab, agarose gel slab, the next step is simply transferring it into a nylon paper or nitrocellulose sheet. So just imagine this is a nylon paper or nitrocellulose sheet. The next step is just this, the fragments are here in the gel, understood, it is embedded in the gel. Now what is done is, this nylon paper or nitrocellulose sheet is made to, it's pressed like this way, so that these fragments come on this. So just imagine these fragments are on this. Okay, now the fragments are transferred on the nylon paper. So all what you need to know is, the next step is transferring of the segregated DNA fragments into a nylon paper or nitrocellulose sheet. That is simply like this. So the diagram also shows like this. This is the agarose gel. Once again I am reiterating, agarose gel, just this, and we get it on this. Okay. This process is known as southern blotting. So our fourth step is southern blotting. So I hope the diagram also is clear, indicating southern blotting. It's simple as this. The segmented DNA or the fragmented DNA are transferred to a nylon paper or nitrocellulose sheet. The next step. So what's the color of the nylon paper? Yellow color. So we see what is placed here, the yellow paper. Next step is very important. It is hybridization of the DNA. The next step is called hybridization of DNA. In this step, what is done is, before the procedure could start, the VNTR of the criminal or the sample obtained is identified. For example, let's assume the VNTR to be A, G, G, C, C. For example, let the VNTR of this person be AA, GG, CC. Now what is done? This VNTR is identified. Now DNA complementary to this VNTR is made in the lab. So what is complementary to A? T. Again A, T. Complementary to G, C. Again complementary to G, C. Complementary to C, G, G. So you can see what is made in the lab, a DNA probe. So the green indicates DNA probe, and the violet, let it indicate the VNTR of the person. There's another technique of identifying the VNTR of that person. So this violet should be the VNTR of the person, and the green represents the DNA probe. So the DNA probe, remember, it is complementary to the basis on the VNTR, not complete. And this DNA probe is labeled with a radioactive isotope. So let me give it some. It is labeled with a radioactive isotope. Now it is ready for hybridization. What is hybridization? These probes, DNA probes, which are complementary to the basis of the VNTR and labeled with radioactive isotopes, are made to bind on the DNA fragments on the nylon paper. So wherever there is VNTR, it binds. For example, I am drawing on this a number of fragments. Let me take blue color. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 fragments. Okay, then on the nylon paper. We don't know where and all there are the VNTR. How it is identified? When we add the DNA probe. Correctly, the DNA probe will go and bind on the places where there are the VNTR. So just imagine on the third segment there was a VNTR. Let the third, sixth and ninth segment there are the VNTR. So correctly the DNA probes come and bind at the third, sixth and ninth segment. See, the third segment they bind, sixth segment they bind and the ninth segment they bind. So the purpose of the DNA probe is to bind to the VNTR. Still it is not visible to the naked eye. So what is done? So before going to the next step, once again I am explaining hybridization. Hybridization is simply binding of DNA probes to the VNTR. Wherever it binds, it will be identified in the next step. So what is done in hybridization? Before we could start hybridization, the VNTR of the person is identified. And I told you here the VNTR of the person, let it be AAGGCC. So that is the VNTR of the person. So a DNA probe is made which is complementary to AAGGCC. That will be TTCCGG. So DNA probe is made, and not only that, it is labeled with a radioactive isotope. Now hybridization occurs wherever there is VNTR. So we have on the nylon paper the different segments. We don't know where there is VNTR. Now the VNTRs are added, that is DNA probes are added, and correctly at 3 where there is a VNTR it binds, at 6 where there is a VNTR it binds, at 9 where there is a VNTR it binds. This is an assumption, I am just giving an example. Okay, so we saw the process is over, hybridization is over, but still not visible to our eye. So to make it visible, what is it? The next step. The next step, again I am continuing with red color, the 6th step is autoradiography. In autoradiography, what is done? This nylon paper is exposed to X-rays. An X-ray unit is used and an X-ray film is developed. And when the X-ray film is developed, you can see wherever the VNTR has bound, it appears as bands. So this is a place where there are bands. So these all represent the VNTRs. And let there be the other segments where there are no VNTRs. So correctly we can see at the third segment, there's VNTR, at the 6th segment there's VNTR, at the 9th segment there's VNTR, at the 12th segment there's VNTR. So wherever there is a VNTR, the DNA probe binds. How did we understand? When the X-ray film was developed, the other fragments will not be visible. I just highlighted the other fragments. There are the other fragments which I have indicated in green, but they do not have the VNTR. So they do not appear in the X-ray film. So I am going to erase it. They are not found in the X-ray film. So I am going to erase the greens. Only the regions where there are the VNTR, it is seen as bands. So we have a banding. This is called the DNA profile. This sheet, the nylon paper, was exposed to X-ray. So now definitely there is an X-ray sheet. This X-ray sheet which shows where the hybridized DNA. What is hybridized DNA? Where the DNA probe has bound with the VNTR is known as the DNA profile. So once again reiterating the steps or the procedure in DNA fingerprinting. There are 6 steps in all. The first step is isolation of DNA. Okay, the first step is isolation of DNA from the sample obtained from the crime site. Here the sample is blood. It can be semen, it can be a vaginal swab, it can be saliva, whatever be the sample. Here the sample that is displayed is blood. So from the blood sample, the DNA is isolated. So this is the DNA that is isolated, and as I told you, it's 2.2 meters long. Then the next step is fragmentation. Using the molecular scissors, restriction endonuclease. So the DNA, next stage, what happens to the DNA? It is converted to fragments of various sizes. The third step is these fragments should be sorted according to their size, and for that we have to go for the step called gel electrophoresis, and we will be dealing with gel electrophoresis in a chapter in biotechnology, where in the well the fragments are fed and electricity is made to pass through the gel, and when electricity is made to pass, it gets separated. The smallest one moves further away, smallest fragment, and this is the largest fragment. Okay, so here all you need to know is the purpose of gel electrophoresis to segregate the DNA based on size. Then fourth, from the gel it is transferred to a nylon paper or nitrocellulose sheet by a process called southern blotting. So this process is called southern blotting. Okay, just like this. Okay, and now you get all these segments transferred onto the nylon paper. Then the fifth step is hybridization. Now for hybridization, we have to know extra points is the VNTR of the person is identified first. And as I told you, the VNTR for example is A 4 times. So the DNA probe can be synthesized. The DNA probe will be complementary to the VNTR. The DNA probe will be T. I made it very simple. Okay. So what is done in hybridization is the DNA probes are put into this, and wherever there is a VNTR, it binds. So if there are number of 10 segments, only wherever there is the VNTR will the DNA probe bind. This binding is called hybridization. So where it is bound, let it be yellow in color. Okay, I am binding at random. Okay. Now this cannot be visible to our naked eyes. So then we have to go for the next technique where this film or this sheet is exposed to X-rays. This is called autoradiography. Sixth step is autoradiography. It is just simple as that. This is exposed to X-rays and we get the film. The X-ray film depicts only certain bands. Not all the fragments are not exposed here. Only certain bands, and these bands correlate to what? The hybridized DNA. That is, the DNA probe that is bound to the VNTR is only exposed as bands. Okay, this forms the DNA profile of an individual. This is the DNA profile of an individual. So once the X-ray is developed, we can see only the regions where there are the VNTR. Why it is shown? Because the DNA probe which is labeled with radioactive isotope has been bound. So that is only exposed in the X-ray film. Clear? This is the DNA profile. So next, I will go to the next slide where we are going to compare. Very important step in forensic science where the comparison is done. So all this is done with different samples of different suspects, maybe 2, 3, 4, 5, 6 suspects may be there in a case. So then the samples are taken from these suspects along with the sample obtained from the crime site. And the last very important or crucial step is matching these profiles. Now I have a slide which is showing the DNA profile of 3 suspects, suspect 1, suspect 2, suspect 3, and this was the DNA profile obtained at the crime site. Now with which profile does this match? Just check with suspect 1, here okay, same, here also almost okay, but this is missing here, then here this is missing, and here it's same, but here it's missing, here it is same. But look at the next suspect. By using another color you can see this is correctly going with this, correctly coinciding, then here it's coinciding. So it's exact replica of suspect 2. Whereas suspect 3 we can rule it out. See it's not at all coinciding, totally different. So the real suspect is this and this. These 2 and 3, suspect 1 and 3 are innocent, suspect 2 is the real culprit. So like this suspects of convicts are caught and they are penalized or sentenced to jail, etc. This can be also not only applicable for murder, rape and theft. It can also be used for settling paternity-maternity disputes. If at all a person claims that so and so is the father of her son or daughter, then we can go for DNA profiling. That we will not call it suspect, we call claimant. These terminologies vary. Claimant 1, claimant 2, claimant 3, and this is the son or daughter's DNA profile, son or daughter's DNA profile, and we are trying to prove the settling the paternity dispute. Okay, so who is the claimant for this child? The claimant father will be number 2 because it's exactly matching. So this is also to solve paternity, sometimes maternity disputes also. Okay, and we can see some more applications. As of now we saw murder, rape, theft, and settling paternity-maternity disputes or parentage. There are other applications of DNA fingerprinting. We'll go to the next slide. So, applications of DNA fingerprinting. First, as I told you, to establish paternity and parentage. Usually it comes with paternity. Okay, so to establish paternity disputes or even parentage. Suppose there's a fire in a hospital and the children in the ward gets interchanged, then how will they go? How are the children brought back to their parents? In that case, we have to go in for maternity, checking of the maternity. Okay. Second case, to identify victims of war and large scale disasters. So, victims of war, definitely they get disfigured, their face cannot be identified, their dead bodies cannot be identified and given to their relatives. Same way in the case of certain disaster. It may be natural or man-made disaster. Sometimes it might be so much disfigurement that the closest relatives cannot identify. In that case, we have to opt for DNA fingerprinting. Third, to determine the population and genetic diversities. It's used in the field of demography also to understand population and genetic diversities. Fourth, as we dealt in the beginning itself, to track criminals of rape, murder and theft. Okay, anyway, if there's a rape or murder or theft, definitely some parts of the victim and the convict will be left behind the crime scene. At least the blood sample, sweat sample, saliva, semen, anything, even the hair follicles can fall. It is a situation where this is done not at ease. It's a quick action process, so something will be left behind. So, no one can go noticed because this is DNA fingerprinting. And lastly, it is used to settle immigration disputes. So, once again reiterating the applications of DNA fingerprinting: 1, to establish paternity and parentage; 2, to identify victims of war and large scale disasters; 3, to determine population and genetic diversities; 4, to track criminals of rape, murder and theft; and finally, to settle immigration disputes. So, definitely DNA fingerprinting, the discovery was a great breakthrough. So, now I will wind up with a small animated video. I think so you will find it interesting because it is a cartoon version.
The burglar smashed the window, leaving blood on the broken glass. Samples of blood, hair or skin can be collected from a crime scene. Forensic scientists investigate the samples in a lab. First, they extract DNA from the cells. They use enzymes to cut the DNA into shorter fragments. The fragments are placed into a well in a gel. They are separated using an electric current. Different sized fragments travel different distances in the gel. The smallest fragments travel the furthest. A pattern of bands is produced. This can be compared with the patterns of DNA from possible suspects. The enzymes cut different people's DNA in different places, producing a different set of fragments. The different sets of DNA fragments will produce different patterns of bands when separated on a gel. Each person has unique DNA, which will produce a unique pattern that can be used to identify them. If 2 patterns match, the samples are from the same person.